This is a production of Cornell University.
For that really lovely introduction. I'd like to start out with some thank yous of my own. Big thank you to my committee, Matt, Lori Drinkwater, and Harold. You guys have been wise and pragmatic and encouraging and supportive, and it's really helped me shape my research. Also, a huge thank you to my lab, the Sustainable Cropping Systems Lab. Seriously, couldn't have done it without you all, and I'm so grateful and appreciative that we work so well together as a team. It's so much fun to go to work every day. A big thank you to Jerry Cherney, who helped with Forage quality, Stephen Mirsky, my USDA collaborator, Paul and Mike out at the farm, Dave Wilson from King's AgriSeed, who helped select seeds for me. Big thank you to Sonam and to the grad students within the department and other plant science departments. So just a really straightforward background, an outline of how the presentation will go, some background information, some methods, and then the results section, which I'll be presenting in two parts. One is crop performance, so a little bit about crop production, and the other one is more applied, so some of the benefits and trade-offs of the different seed treatments I use. And then we'll do a summary, and then I'll tell you about some future plans I have. Matt didn't say this, but I will be doing a PhD with Matt. I couldn't imagine leaving the lab, this winter or anything. So just to start off with, this is a graph that describes the trend in dairy farms distribution. So it's the number of farms over time is declining, and the average farm size is growing. To me, this trend is very disturbing. It's not just for dairy farm, it's true in other ag systems. But particularly within dairy farms, there's been this huge decline. 200 farms with less than 200 cows have, the farm size has decreased between 2000 and 2006 by 30%. So that's really huge. This trend is predicted to increase. There are a lot of structural differences that come with farm sizes, and probably the biggest takeaway of that for this presentation is that smaller dairy farms tend to grow more their own feed. This is a map that describes forage production in the United States. So for those who aren't familiar, forage is the edible plant part that isn't separated. The grain is not separated, and the forages are then fed to grazing animals. So cows, sheep, goats, but in this case mostly cows. New York is a huge producer of forage, third largest producer of corn silage, alfalfa and timothy also up there, and most of those forages go into dairy production, and New York is the third largest producer of milk in the country. This is a picture of the 2012 drought in August. I started grad school at this time. So I started when this drought was happening and when I was actually studying agriculture for the first time. And it was very influential for what my research eventually came to be, but it was also it was huge. It happened in the States, and 80% of agricultural land was affected. 75% of corn and soybean production were severely or greater affected by the drought, mostly in the heartland here, but also you can see that New York State up here, it has some different colors. It was affected by the drought, and it was this that kind of took my research in a way on a certain path, and it was very emotional for people. I think we would drive past and we would see the corn that was just very stunted, or we would see farmers standing in their fields just looking lost and desolate. They had no idea what they could do. And so one of the options farmers have is that they could plant these summer annual forage intercrops. So typically, right now, they're mostly known as these emergency forages, and they're planted after corn failure, but they also fit into the small grain system. So that's sometimes known as triple cropping, and that's when you have a winter cereal and it's coming out. So, you know, like after triticale or after wheat, and then you plant these things in the middle of the summer, and then the growing season is short enough that you can get them out in time to really establish another cereal crop, another grain crop into the winter. So you can plant these between the middle of June and the middle of July. They're pretty fast growing. You can get a forage cut between 35, 45 days. The seeds reach maturity around 90 days. And the species I've selected for this presentation for the research, these are drought tolerant and in general require fewer inputs than corn silage. There's some very basic benefits for intercropping. One, you're decreasing the risk by planting multiple species. So by having different species that react differently, that use the resources differently, you're spreading out that risk and you're really tapping into the functional diversity of the plants. This, in turn, increases the resilience of the ecosystem, and it strengthens it because you really are depending on the species that are there and the things that come with it by changing the pest or weed pressure, changing the microbial communities of the soil, and instead of relying on these external inputs, which I think is quite a fragile system. And so this is also, Pearson calls this the regenerative semi-closed agriculture. So it's when the nutrients are kind of coming off of the farm, and then if you have a dairy production, that manure is staying on the farm. So if you think about it in terms of where the nutrients are going, it's a more closed structure than if you're exporting nutrients to and from. This is so some related summer annual intercrops. I think there's a growing interest among farmers, and certainly the NRCS has been promoting these summer annual intercrops as cover crop cocktails and mixtures. This is Ray Archuleta from the NRCS. I think we should make a motivational poster of this for the lab. Is this NRCS guy directing us to plant summer annual intercrops? There's been research done by Kevin Armstrong in 2008. They were comparing grass legume mixtures for forage quality for with corn silage, and they tested out some different legumes, and they said that the lab bean increased the forage quality because you're increasing the crude protein, but you're not decreasing the yield. So that's pretty key for farmers. You don't want to take a yield cut. Dan Brainerd also did some research with Robin Belinder regarding grass legume mixtures. They chose 2 grasses and 2 legumes, and the best combination they found was cowpea with millet, which improved nodulation. It lowered the seed costs because the grains are less expensive than the legumes, and also they managed to reduce the weed seed production than if they were planting the cowpea separately. So some considerations with intercropping. I find intercropping experiments, they're very challenging because they get so big so fast, because you're like, what species should I look at? Oh, and I should look at the seeding rate, and all of a sudden you have like 500 different treatments, and then your committee says, probably shouldn't have 500 different treatments. So species selection is very important, and even within species, different cultivar selection is important. The seeding rates you choose, and then the ratio of those seeding rates to the other species, as well as the soil fertility. So, you know, depending on your playing field, where you're starting off with, really, what am I trying to say? It really affects the crop production later on. So if you have very fertile soil, then perhaps the grasses would do better. But if you have more legumes and less fertile soil, then the legumes would do better. So just to summarize my entire 2.5 years here in one slide, I'm testing the ecological theory and quantifying potential benefits from intercropping. And I'm doing this within an alternative warm season, drought tolerant, functionally diverse cropping mixture in the Northeast and in the Mid-Atlantic. And I'm also, so these 2 points, these are like a little more theoretical, but I'm very much interested in how does this knowledge apply to farmers? How can people use this information. And so I really want to quantify the trade-offs between forage quality and yield and determine optimal management. I really would like to tell farmers at the end, I've tested this and I think this is the best mixture or the best thing you can do in your situation. So I have two main research questions. The first is, how do crops reform, and are there even benefits to intercropping? And the second research question is, what are the trade-offs between yield and forage quality over time? For species selection, I chose 4 species and I based it off of 2 different functional traits. So nitrogen fixing ability, whether a plant is a grass or a legume, and also plant architecture. So the stature of the plant, whether it's bushy or taller. And so for the 2 grasses, I chose a hybrid pearl millet, and it's abbreviated with an M, and it's throughout the presentation. And it's the shorter variety of millet used for forage. And then for the tall grass, I used sorghum Sudan grass. It's a BMR variety, and it's a cross between sorghum and sorghum Sudanese grass, and it looks very similar to corn. It grows quite tall. For the 2 legumes, I used cowpea. In the South, we call them black-eyed peas. They're abbreviated with a C. And then sun hemp. And sun hemp is abbreviated with an H. And sun hemp, I think, is the least familiar of these crops to this audience. It's native to India. It's this fiber and forage crop that they grow there, and it's tall. It grows quite tall, and it's very beautiful, and the leaves and the stem are iridescent. So I had 2 growing seasons, 2013 and 2014, and 3 different field sites. We were at Beltsville, Maryland, the USDA headquarters, and then Aurora, New York, which is about 40 miles away from here. And then our most northern site was in Willsboro, New York, and so that was in the middle of the Adirondacks next to Vermont. And we had a different number of fields, so if there was ×2, then we had 2 fields at that field site. And we planted between the middle of June to the middle of July. We had 4 to 6 reps per site. For the experiment design, we prepped the seed beds. This is Chris on a tractor. So we prepped the seed beds, plowed, disc, cultipacked. We applied 35 kilograms per hectare of nitrogen in the form of poultry litter. And 35 kilograms is half the recommended amount as sorghum Sudan grass. And the idea for doing it like that is that if we fertilize at the full recommendation, then we're really shifting that fertility, that competition over to the grasses, which are able to use that nitrogen more efficiently than the legumes. And then if we didn't fertilize at all, we were afraid we would shift the competition too much to the legumes. And so we split the difference and decided 35 was a good number. No pesticides or herbicides were applied. I think that's pretty typical for a forage system, but this is low input, so I would like to emphasize that. And seeds were drilled to 4 centimeters down. And plot sizes range from 9 to 40 meters squared. This was a single cut, so in some forages you can take multiple cuts over time, and then you take cuts of the regrowth. But because we're using a mixture of species, they have different maturity dates, different regrowth rates, we just did a single cut system. And that was also a way to keep the number of samplings down into the pragmatic sphere. For seed treatment, so we have the 4 species in monoculture, and these are their seeding rates here in this middle column, and again the abbreviations, and then the cost per hectare of those seeds. And then we tested 4 3-species mixtures, and they're indicated with an X. And then we have their cost per hectare, and of course the one possible 4-species mixture. The seeding rates were, for the 3-species mixture, each species was seeded at 1/3 of its rate in monoculture. So if you're looking at millet in a 3-species mixture, 21 divided by 3, you're seeding at 7 kilograms per hectare. And then for the 4-species rate, you're seeding at 1/4 of its monoculture rate. And it was a randomized complete block design. Different sampling methods. So we took crop density, crop biomass, and weed biomass. We essentially laid a 1/2 meter square quadrat down, and we clipped at 10 centimeters above the soil. This was to simulate a forage cut where they're not going to be clipping right at the soil level, but a little bit above. And then crops were separated out by species, and then dried, and then later weighed. We had 2 sampling dates in 2013, 45 days and 90 days after planting. And in 2014, we added an additional sampling date at 35 days. And so that was the purpose of that was to really get to the forage quality question. How does forage quality change over 35 and 45 days, which is kind of the sweet spot. In addition to the biomass sampling, we also took light transmittance to assess the canopy cover. We did that by sliding a line sensor underneath the canopy and then holding a point sensor above the canopy and then taking simultaneous measurements, and that was to capture how much light was passing through the canopy. And also we did take some soil samples to understand the baseline fertility of the soil that we were working in. For data analysis for this presentation, I'll be talking to you about the 45 days pooled across sites and years. There weren't major site differences. All the orders of the treatment were pretty similar across all the sites. There were really big year differences. I don't know if you all noticed, but 2014 was quite a bit colder than 2013 for the summer. So the biomass production in 2014 was almost half of that in 2013. To analyze the data, we did an analysis. I did an analysis of variance with treatment as the fixed effect, and year, block, and location were considered random effects, and then I used a 2-key test to show the differences between the means. So this is the results section, part 1, the crop performance part. And this is a couple of shots of us in the field. This is last year at Aurora, about a month after planting. And the letters here indicate the seeding treatments. So you can see the cowpea and monoculture, the canopy really hadn't closed, especially if you look at the 4-species mixture. And then 2 months later, you see here the millet and the sun hemp were planted in monoculture. They're looking very full and nice, but when you look at the species mixtures, they just have so much texture. They just look like they're taking up so much more space. This is data for the 45 days after planting pooled across sites and years, and this is the total biomass. And down at the bottom we have the 4 species in monoculture, the 4 3-species mixtures, and then the 1 4-species mixture, and then the crop biomass in kilograms per hectare on the y-axis. And you can see that the legumes in monoculture did not really do that well. The grasses were very similar to the mixtures, with millet, sorghum, sun hemp producing the highest amount of biomass, and then the sorghum, sun hemp, cowpea producing the lowest amount of biomass in the mixtures. This is a graph, the same one you saw before, except now you can see the composition of the mixtures. And so each color stands for a monoculture, and then here you can see a lot of purple and green, and those are the grasses, and they make up quite a bit of that crop biomass. Dominant weeds. We had some issues with volunteer crops depending on our field sites. So at one site in Willsboro, we went in after a canola variety trial, and there was a lot of canola. But, so it was like very terrible because we considered it weed biomass. In reality though, that stuff makes great forage. So Mike kept trying to convince me, oh, you should run the the forage quality analysis with the weed biomass. And I was like, yes, I would love to do that. That's, like, we know this would be so much higher. We also had some issues with some volunteer wheat. So at Aurora, we had a later planting date, and I think Paul was driving his tractor, driving the combine off the field as Chris was driving the tractor onto the field to prepare the weed seed bed. So any wheat that didn't make it onto the combine had a very nice wheat seed bed to grow into. But again, it would probably make for some decent forage quality. Another field, we had an issue with mustard. I'll talk a little bit more about that in a second. And then pretty much the usual suspects of summer annual weeds: ragweed, pigweed, lamb's quarter. So here we have a weed phytometer, and this mustard in bloom was beautiful. It really was beautiful, and it was great also for seeing the treatment differences. So you can just look here, and I've divided these fields, this field by the different treatments. You can see the mixture that contained the sorghum was very good at outcompeting the mustard, but this very dark yellow, lots of yellow down in this corner, this was a cowpea monoculture. So it really, you could just tell the difference of how competitive some species and some mixtures were. This graph shows crop biomass in the brown and the weed biomass in the yellow bars on top. So you can see that the legume monocultures produce the most weeds. Grasses were not very weedy. Some of the mixtures didn't have that many weeds. They were all a little similar though. And as terrible as the cowpea and sun hemp were at suppressing weeds, they certainly did a good job, more than they could have. In 2014, we had a control plot. We put control plots in all of our fields. You can really tell like how much less weeds there are, even when just something was planted. So in addition to the crop production and the crop biomass and the weed biomass, also doing some additional metrics: land equivalent ratio, which measures the efficiency of mixtures relative to monocultures; the crop growth rate, which just shows the change in biomass over the change in time; and also species evenness by biomass, which shows the relative production, measures the relative production and species dominance. And so these are all like three kind of snapshots of how these mixtures performed and how the species within the mixtures performed. The first one, land equivalent ratio. It is the sum of the ratios of intercrop yield for a species divided by the monoculture yield. And I've tried to depict it in this cartoon here. So in the intercrop, you can see that all the species grow better than when they're planted separately in monoculture. And this is due to less intra-specific competition. So LERs that are greater than 1 describe when intercrops are more efficient than monocultures. And very excitingly, all the mixtures that were planted for this experiment had LERs that were greater than 1. So all of these mixtures were more efficient in terms of biomass production per space than if the species had been separated out and planted in monoculture. This is crop growth rate measured in kilograms per hectare per day, and here we have 2 columns for the grasses and 2 columns for the legumes. And I showed the slope up here. You can see the crop growth rates for the grasses in monoculture are really high. And so it shows like a much steeper slope than the legumes, which have a lower crop growth rate, and they have a smaller slope. So I just want to walk you through this one. Millet's growth rate, when it was in monoculture, it was about 17 kilograms per hectare per day. And then when it was planted in these mixtures, it decreased. In this mixture, it didn't decrease significantly, so the growth rate when it was planted with the sun hemp and the cowpea was similar to that as if it were planted in monoculture. And the other growth rates decreased a half, maybe a third. A similar trend you see with sorghum. It has a very high crop growth rate when it was in monoculture, and it falls when it is planted in mixture. So remember, you're also seeding at like 1/3 to a quarter of the monoculture rate. The legumes are a little bit of a sadder story. So their crop growth rate in monoculture were lower, just in general, than the grasses. So these plants, characteristics, just have a slower growth rate. And when they were planted with mixtures, they just got whomped a little. Cowpea fell by decreased, the crop growth rate decreased by an order of magnitude, and so it was really facing a lot of competition when it was intercropped. So the third snapshot, the third additional metric I want to describe to you is species evenness. And usually species evenness is done with plant density. We thought that doing it by biomass was a little bit more revealing. Here we see low evenness in this cartoon, and you can see that the one species is much bigger than the other species. And then a high evenness means that the plants grow similarly well. So species evenness is a metric that goes from 0 to 1, and the blue bars here indicate the evenness at 45 days, and the brown bars are evenness at 90 days. And so you see that millet, sorghum, sun hemp, it was the most even species at 45 days and also the most even at 90 days. And that just says those three species, when they're planted together, if you think of them as a team, each teammate is holding its own weight a little bit more than if you had sorghum, sun hemp, and cowpea. You can just imagine the cowpea was dominating. Its growth definitely was much greater than the sun hemp and the cowpea. Just in general, species evenness decreased over time. So as the growing season went on, there was a decrease as one species became more dominant than other species in some of these mixtures. So to summarize this part of the results section, mixtures produced a similar amount of biomass as grass monocultures. There was over-yielding seen by all the mixtures, indicated by an LER greater than 1. And there is a higher crop growth rate in general in the monocultures, except for millet with sun hemp and cowpea. Mixtures with 2 grasses tended to be more even, and mixtures with 2 legumes tended to be less even, and there is a decrease in evenness over time. So now I want to switch to the more applied findings, the second results section, and this goes back to the question: what are the trade-offs between yield and forage quality over time? And so just a very short primer on forage quality. Forage quality tends to decrease over time as the plants get larger, while the yield tends to increase. And so that reason mostly is because when the plants are younger, they have a lower fiber content. And that fiber content means the animal can consume more of that diet, and also it's higher energy because it's full of the cell contents are full of these nutritious, digestible, quickly digestible things, sugars, proteins. As the plant ages, there's an increase in these structural carbohydrates that make up the cell wall, particularly hemicellulose and cellulose. And so when it's a high fiber diet, when the plant is older, then there's a lower intake by the animals because they feel fuller. They have more fiber when they're eating that, and because they cannot consume as much, there is a lower energy just in general of the diet. So this is relative feed value at 35 days in 2014, and relative feed value is this metric between digestibility and fiber content, and it was used by the industry for a very long time before it became affordable to test for the protein content. I was not able to get the protein analysis done in time for this presentation, so I'm using this metric even though it only tells part of the story because it doesn't require any protein content. So relative feed value, relative feed value is this metric that's based off of forage legumes, and it gives you this one number of how good or not a forage is. And so we can see the relative feed value is quite high for the legumes, and this metric tends to underestimate forage quality for grasses, so that's the reason it's not used as much anymore. And so the grasses in monoculture are quite low. Then we have these mixtures. The millet sun hemp cowpea has the highest forage quality of all the mixtures, and the other mixtures perform similarly well. These yellow bars here indicate what kind of feed you would like to give to a cow. The brighter yellow is the kind of quality of feed you would give to a lactating cow, and the less yellow, the browner bar, is feed that's still good enough, but maybe not as much energy that you would give to a cow that is producing milk. So you can see that mostly, except for the sorghum, just by a little bit, all fit into this range that it's safe to feed and even good to feed for lactating cows. And again, this metric really underestimates the forage quality of the grasses. This next graph really tries to answer the question of what are the trade-offs between yield and the forage quality. And so the best way to do this metric was just to multiply the relative feed value by the yield. So we know that the grasses had a very high yield compared to the legumes, but we also know that the grasses didn't have that great of a forage quality. And here we see these big differences. So the purple bars are the first harvest at 35 days, and then the green are 45 days, and the blue are 90 days. We see some really nice high bars for these legumes when they're in monoculture, and that falls. And part of its falling is due to a decrease in forage quality over time, but part of it is also to the fact that they don't yield quite as much as the millet or the sorghum. So you have yield that's really pushing up these bars here. I didn't want to overwhelm you all with all the bars all at once, but here are the different mixtures. You can see that the millet sun hemp cowpea had the highest forage quality for the mixture. It falls, and then yield really takes over for some of the yield is making up the difference in the bar sizes for some of these other mixtures. So just in general, I want to summarize that there were a lot of site year differences for forage quality. Forage quality decreased over time. Optimal harvest for the grasses and the mixtures using the relative feed value metric was at 35 days. Legumes contained less fiber and were more digestible, and grasses in monoculture had a higher fiber content. And because most of the mixtures, the majority of that mixture, the biomass of that mixture was grasses, the mixtures also had quite a high fiber content as well. Here I would like to touch on the economics. I'm really excited to develop some of these ideas more, but But here we have the cost per hectare that we saw at the beginning for the seeds, for the different seed treatments. We have 35 days, 45 days, and 90 days production per dollar, so that's kilograms per dollar. And you can see really that millet has this very low seeding cost per hectare, and this is for a couple of different reasons. One, but related reasons. The cost of millet is so much lower is because the seeding rate in general is so much lower. It was 21 versus 60 or 80 for some of the others. And the seeds for millet are quite a bit smaller than some of the other seed treatments. So you can plant less of it, it's less expensive. And it's really driving the trend here for the production per dollar. This is also seen here with the millet, sorghum, sun hemp produced the most, and it had the highest production per dollar. I think one of the challenges that I've seen when doing this research is that the seed costs to me seem pretty prohibitive for farmers to actually do, but I think part of that is because of the seed economies of scale. So if more people adopted this practice, it would drive down the price, that you would have more suppliers entering the market and producing varieties that are high yielding for this region at a lower cost. And also, forgot to mention this, the 4-species mixture came out right in the middle between all the 3-species mixtures. So some final recommendations. We looked at a lot of different metrics. We looked at yield, we looked at weed biomass, weed suppression, and we looked at the forage quality. We touched on the cost. So there are a lot of different factors in play as to what I would recommend farmers to do. Just to summarize, millet had the greatest production. It produced the most biomass, and it was the lower cost by far. Millet, sorghum, sun hemp mixture had the greatest production of a mixture, and it also had the lowest cost per mixture. It had a really high LER and a really high evenness that was saying, the sun hemp wasn't being completely dominated by the millet or the sorghum. And the RFV, the relative feed value, was near that threshold for lactating cows. So it was the highest producing, perhaps with a better forage quality metric, it would put it into that range of being a good forage quality. The millet sun hemp cowpea mixture had the greatest forage quality, was also the most weed suppressive, and in that mixture the millet had the same growth rate as it did in monoculture. So you're not taking a yield loss if you were going to have the mindset of planting a grass with some additional legumes. You weren't taking a yield loss there, but it had a little bit of a higher production cost. To close, I just want to talk a little bit about some of my PhD work that's also had 2 growing seasons, and maybe some of you were wondering, what about those bicultures? Well, don't worry, they're coming. So for this work, for these experiments, which are also called SAFE, the Summer Annual Forage Experiments, I'm looking at monoculture rates, so that recommended seeding rate and how it changes if you're planting, how the biomass changes at 70% of the seeding rate or 90% of the seeding rate, and also looking at all the biculture combinations. So there are 4 species, there are 6 possible biculture combinations, and more than that, more than just looking at like 50-50 of millet and sorghum and 50-50 of millet and sun hemp, I'm really looking at different seeding rate ratios, so like 20% millet and then like 60% sorghum, and doing that for all 6 combinations of bicultures to really understand what are the dynamics within the seeding rate ratios and how does that relate to the recommended seeding rates of these species in monoculture. And so then I will be doing some modeling of the competition between the intercrops, really getting at intraspecific competition and interspecific competition. And finally, for some other future work, I'm going to be doing some farmer interviews, understanding metrics. I'm really interested in understanding the metrics for behavior change in terms of agricultural production. So what policies need to be there that would influence farmer decision making, what barriers cannot be there. I think this is incredibly important and is something I'm very interested in. It's in that expansion stage of a PhD where you're saying yes and this, and yes and this, and it hasn't been condensed yet. But in general, I'm so interested in science communication, the relationship and the communication between scientists and farmers and scientists and the general public. I'm also interested in agricultural policy, potentially after a PhD, maybe going to Washington. So with that, I will take some time for questions. Yes?
This has been a production of Cornell University, on the web at cornell.edu.